Okay, so moving into section 15.2, we're going to take a look at the extracellular matrix. So we're outside of cells here, so we're not talking about the insides of cells, we're talking about materials which are produced and function outside of the cell, outside of the plasma membrane. And what I'm going to say here is going to vary a little bit with what you'll hear in anatomy and maybe physiology, but it just helps give us kind of a framework to work in for cell biology. We can think about three basic or fundamental types of extracellular matrix, or extracellular material as it's sometimes called. We can think about really dense ECM, like bone, and that's consisting mainly of a rigid extracellular matrix, and that contains a small number of interspersed cells. And we think about cartilage, which is not as dense as bone, obviously, and that is constructed mostly of matrix material that is more flexible than bone, given the function of cartilage. And then we think about connective tissue just generally. Now, I know that bone and cartilage and blood are all types of connective tissue, but we're going to think about bone, cartilage, and then connective tissue, and we can think about loose or dense connective tissue. So I'm just going to think about three fundamental types of extracellular matrix. So I'm not telling you that these are the three types of connective tissue. I'm telling you that they are characteristic, these three, bone, cartilage, and connective tissue, are characteristic of three fundamental types of extracellular matrix. So there's a difference between what I'm telling you here and what you may interpret what you're told in anatomy or physiology. So connective tissue there, that's going to be surrounding glands, blood vessels, that kind of thing. It's relatively gelatinous, and it contains cells, things like fibroblasts. It can be penetrated by macrophages, that kind of thing. So despite the fact that we've got three very different types of tissue here, three different types of ECM with diverse functions, we can generally think about three kind of classes of molecules that you always see in these three types of connective tissue. Structural proteins, things like collagens and elastins, which provide strength and flexibility. Protein polysaccharide complexes, which are kind of complicated, called proteoglycans, that provide the matrix kind of structure. And then adhesive glycoproteins, things like fibronectins and lamins, that allow the cells in the extracellular matrix to attach to the matrix itself. So those three kinds of classes of molecules. Let's think about them kind of in turn. Let's start off with collagens. Probably the most abundant ECM component in animals is collagen. And so this is actually a family of proteins. And it's secreted by several types of cells in connective tissues, including fibroblasts. And now the structure is kind of interesting. You can actually see collagen fibers in the extracellular matrix when you look at ECM through an electron microscope. And so you can see these collagen fibers here, and collagen fiber is made up of numerous fibrils, and each fibril is made up of collagen molecules, three collagen molecules wrapped around, and the three molecules are called alpha chains, and they wrap around one another. So if you think about you know those big kind of like thick ropes or steel cables you get on docks? This is very much like that, given its composite nature. And so they all occur as this rigid triple helix of intertwined polypeptides. The weird thing about them is they've got some really weird amino acids. They contain a lot of glycine, and they also have two unusual amino acids, hydroxylproline and hydroxylysine. And the glycine is an amino acid you often find when you actually create helices, and so that contributes to the helical structure of these alpha chains. So think about how you get this strength from the composite nature of these three intertwined alpha chains making up the fibrils, and then the fibrils being composited together to make the fibers. Think about how much strength that would give you. Collagen synthesis is kind of interesting, because part of it occurs in the ER lumen, and then part of it occurs after secretion outside the cell. So in the lumen of the ER, three chains, you have synthesis of individual chains, obviously, and and so that would be an alpha chain, and then 3 chains assemble to form a triple helix, and that's called procollagen, and that all occurs in the ER lumen. So that's a nice example of multimeric protein, multimeric subunit assembly in the ER. And then that gets processed through the endomembrane system and ultimately secreted, and once it's secreted, then the collagen molecule is produced by being trimmed by a procollagen peptidase. That's an enzyme which kind of trims these scraggly ends off, those loosely packed ends off, to give you this nice tightly packed bundle. And then these alpha helices associate, and these 3 alpha helices assemble into a collagen fibril, and then the fibrils assemble into a fiber. And obviously you can't make this collagen fiber inside the cell and secrete that, so you secrete the pieces and then you do the rest of the assembly outside. It would be like trying to build the Millennium Falcon in your garage. You can do some of it inside. You know, Millennium Falcon is right, it's the Star Wars thing. They said Star Trek. You can build some of it inside, but to finish it up, you've got to take the pieces outside and then assemble it outside. If you don't like Millennium Falcon and you're on the dark side, then Death Star, whatever you like. Okay, so that's collagen. What about this flexibility and elasticity that you find in extracellular matrix? Because if you think about compression, when compression happens to your body, that's being absorbed partly by extracellular matrix, and when things can bend and flex, that's also partly due to extracellular matrix. So the flexibility and elasticity comes from these stretchable elastic fibers made from a protein called elastins. So these are proteins which are rich in glycine and proline, and then the molecules are cross-linked by bonds between lysine residues. So you take these individual kind of squiggly coiled up proteins, you cross-link them with these bridges at these lysines, and that kind of gives you like a have you ever seen those slinkies? You know how like the slinky can be stretched out and then you let it go and it kind of comes back together again. You can stretch it out and you let it go and it comes back together again. That's what this is like. And so you have this because you've got this kind of network of proteins which can basically be pulled and stretched apart. So tension causes the overall network to stretch, and then release causes a release of tension, and the individual molecules relax back to this kind of low energy state here. So those are elastins. Last thing we need to look at are the proteoglycans. So the collagens and the elastins are embedded in this network of proteins and sugars. The sugars are attached to the proteins, and that's called proteoglycan. So that's like the real matrix in which you put the proteins collagen and elastin. So you think about that as like a gel-like network or mesh, if you like. And so we have the collagen and the elastin embedded or enmeshed in this gel-like network of proteoglycans. And these are a form of glycoprotein, that's protein with sugars on it, with a lot of a molecule called glycosaminoglycans. Some of those just rolls off the tongue. So I'm going to just call those GAGs. So glycosaminoglycans, or GAGs, are large carbohydrates, and they are made from repeating disaccharide units. So what you do is you make a polymer. You make 2 sugar units, stick 2 sugars together, and then you join those all together. So it's effectively a polysaccharide. And so these are molecules like chondroitin sulfate, keratan sulfate, and hyaluronic acid. So here's a hyaluronic acid, or a hyaluronate molecule. So that is a repeating disaccharide, and that's what the 2 sugars look like. Now don't freak out and worry about the sugar chemistry here, okay? You just need to know they're repeating disaccharides of sugars, and on some of the sugars there are sulfates. So the sugar chemistry is kind of complicated. I don't know it well myself. It's not that important. All you really care about is you've got these repeating disaccharides of sugars, things like N-acetylglucosamine and things like that, and galactose can be thrown in there, glucuronate. So all kinds of different sugars can be used to form these chains of disaccharides, so like hyaluronic acid and chondroitin sulfate. So then you take those sugars and you bind them to these core proteins, And that gives you a molecule called a proteoglycan. So a proteoglycan is like this thing down here. So this one down here is a core protein, and it's got all of these keratan sulfate molecules. So this would also be a proteoglycan up here, and this would be another one up here, and then this would be one here as well. And then they're all kind of connected by other gags as well. So these get attached to the membrane of cells. So different ways that we're not going to worry about, that these can be attached to the membranes of cells. So this forms this kind of like gel-like substance into which we then embed the collagen and the elastins. So this is like the matrix, the supporting structure in which we put the collagen to give us strength and the elastins to give us flexibility. So we'll wrap it up there. We'll come back with the second part of the extracellular matrix shortly. I'll see you soon. Bye for now.